So we've already talked about the structure of DNA. How is it shaped? How are those molecules or those monomers put together to make the bigger structure, the bigger polymer? And now we're actually going to talk about how do we replicate DNA. And replication simply means to copy. We also use the word double or duplicate a lot, which all means the same thing. We're making more DNA. So you really want to think about, if you start out as one cell, how did all of my cells get a copy of the DNA? Well, we have to go through this process. So because DNA is so important to all of our cells, when a cell divides, it must pass on an exact copy of the DNA to its daughter cells so that it can function. And remember that this was important. Those daughter cells could have been gametes or somatic cells. So this is important for making our sperm or egg, or making our body cells like our liver or cheek cells. So because it's so important, we need to copy DNA or replicate DNA during interphase. So if you look at this picture that you have in your notes here, what is this a picture of? Okay, this is a picture of the cell cycle. Remember that the main steps to the cell cycle, or the longest part of the cell cycle, was interphase. This picture is a little different because it has a picture of interphase on the inside and it's going all the way around here. Okay, that's all interphase. And during interphase, our DNA is specifically replicated or copied during the S phase. So this is the section that we are concentrating on, this S phase of interphase. Interphase is made of 3 parts, G1, S, G2. We're really worried about the S phase. The G's, not so much in this case, okay? And do you notice that after this happens, mitosis can occur? So after DNA is, or when DNA is replicated in the cell cycle, we have several steps that occur, and it's actually 4 kind of main steps. The first step to DNA replication is that you have an enzyme, which is actually a type of protein. You want to keep that in mind, kind of a little review as we go. Breaks those hydrogen bonds. What did we talk about the hydrogen bonds yesterday? Were they weak or were they strong? Hopefully you remember that hydrogen bonds are weak. It's important that they're weak so that they can be pulled apart. So between the paired nitrogen bases, this allows the DNA to unzip as the 2 strands move apart. So here is your regular DNA double helix. Whenever we're about to replicate it, it'll actually unwind and form into a ladder. When that happens, between those A's and C's and G's and T's, between those nitrogen bases will actually pull apart and the DNA will start to unravel on both sides. Almost like if you twisted up your shoelace, you start pulling it apart at the bottom, here are the 2 strands that are separating. The reason why we want it to separate is that we're actually going to use the side that's open as a template, okay? So you open up DNA to use as template. A template is just like a pattern, and you're just following the pattern. So the newly unpaired nucleotides, here are the unpaired nucleotides because we pulled apart the A's and the T's and the G's and the C's, will actually bond with free nucleotides that are present in the nucleus. So you have some extra nucleotides that your body has stored, and those will actually bond up. This process is catalyzed by another enzyme. So during all of these steps, you have enzymes that are breaking this apart, enzymes that are unwinding it, enzymes that are putting them back in. So once you're done unzipping, if you notice in step 2 of that picture, you can actually see a new strand. Strand Y is the new strand. New. because now we have new A's and T's and C's and G's that are bonding up with the ones that have been opened up. Then, after we start bonding up the separate sides, the third step is that we'll link the nucleotides along the newly constructed side of the DNA ladder by bonding sugar to phosphate factor. So basically you have a structure. You have your A here, your C here. You have this nucleotide that came with a T and a G. But now we need another side to it. So now the end of the ladder will actually form up. And we'll draw a better diagram of this in class so that we can understand this a little more easily. But the last and final step is that the DNA is going to be proofread by enzymes for any errors. Because what would an error mean? An error in DNA, if you remember from our last section, would actually be called a mutation. And while most of the time, or while not always, mutations aren't always bad, but sometimes they could be. So to just double check ourselves and to keep ourselves safe, our cells will actually check for errors, and enzymes will do that job. After we proofread everything, the DNA ladder will actually twist back up and we'll have 2 new strands. So the end result of DNA replication, 2 identical DNA molecules have been produced. Each daughter DNA, just like how we had daughter cells, copies of each other, is composed of 1 old strand and 1 new strand, thinking about those separate sides. The term for this is called semi-conservative. If you're being conservative, you're being safe, or you're saving something, so you're saving that old strand. Now each copy of the DNA is packaged as a chromatid on a doubled chromosome. So remember that that structure looks kind of like this. Okay, that's your doubled chromosome. And after mitosis, each daughter cell will receive 1 of the 2 copies of DNA, and this happens when the doubled chromosome is split. And that's pulling apart that doubled chromosome. Right now, what I want you to do is to actually take 5 minutes, look back over your notes on this section, pick 2 main terms or concepts, and to write 2 sentences about those 2 concepts, okay? Don't follow what's on the directions on the board right here, but 2 sentences about 2 main things on our notes. You can start all the way back at page 43 to 45.